Good day, our audience. I'm pleased that I'm invited by the organizer to share our experience on this topic, using novel tools of synthetic biology to understand complex diseases via functional genomics. So to start with, let me introduce or give you a bit of background about complex disease or complex trait. So it refers to a phenotype that is contributed by genetic risk factors, environmental factors, and also possibly their interactions. Environmental factors here can refer to diet, lifestyles, education, so on and so forth. Okay, and in fact, complex diseases mostly are common diseases. So, for example, diabetes, hypertension, age-related macular degenerations, myopia, blood lipids concentration, so on and so forth. They are complex diseases and complex traits. Myopia here are highlighted in red because I'm working on myopia myself. Okay, so what is the most powerful method to identify genetic risk factors associated with complex diseases? And we know now, in fact, genetic assurance study are the most powerful approach to find the genetic risk factors associated with complex diseases. So it can be population-based control study or family-based association studies. And because of the ease of subject recruitment, population-based control study is the most commonly used approach. Here, you have a group of subjects with the disease, what we call the cases, and a group of subjects without the disease, what we call the controls. And then you compare between these two groups, the allele frequencies or genotype frequencies. And genetic assurance study is culminated with GWAS, genome-wide assurance studies, which is a hypothesis-free approach. Here, you're going to test up to, say, millions of SNPs or single nucleotide polymorphism or variants in a single experiment. And with appropriate statistical testing, then at the end of the study, you're going to generate hundreds to thousands of SNPs or variants which are associated with the phenotype of interest. So to start with is a hypothesis-free approach, but at the end of the study, we generate many SNPs to follow up. So it's hypothesis generating to end, although at the very beginning it's hypothesis-free. GWAS, in fact, requires very large sample size in order to give you reproducible and robust results. And hence, GWAS are very time-consuming and very expensive. In order to reach extremely large sample size, usually researchers collaborate with each other to pool their data, analyze the data by meta-analysis. So by pooling data together, the sample size can go up to hundreds of thousands or even millions. So that's the usual case in order to get robust results in GWAS, although there is a few exceptions to these generalizations. For example, in age-related macular degenerations, the identification of complement factor H associated with age-related macular degeneration only requires less than 100 cases and 50 controls, which is exceptional because the odds ratio of the SNP of interest goes beyond 7 under the recessive genetic model. For GWAS associated SNPs for a phenotype of interest, so remember GWAS is a type of associated study. So, but then association does not equal to cause and effect relationships. The majority of associated SNPs are non-coding, meaning these SNPs are found mostly in regions outside the exons, in introns, in flanking regions of the genes, or in intergenic regions. Okay, and because they are not in exons, so people are thinking that the genuine causal SNP associated through GWAS are most likely involved in regulating gene expressions. Okay, what's more interesting is the associated lead SNP or the associated sentinel SNPs, meaning the SNP that give the lowest p value in the association study. These lead SNPs are usually not the causal SNPs. And in fact, other SNPs that are in strong linkage disequilibrium with the lead SNPs are also equally likely candidates for as causal SNPs. linkage disequilibrium is a genetic phenomenon in which alleles of different loci on the same chromosomes tend to associate with each other more frequently than expected. LD phenomenon actually help us a lot when we want to identify the SNPs associated with complex diseases. But however, when we zoom in to find the associated SNP, once we find the associated SNP, and we want to find the causative, then LD phenomena actually give us a lot of troubles. Okay, and because of this, then to follow up, then you're talking about tens to hundreds of candidate genes to follow up in order to find the causal SNP. So in fact, this is a bottleneck of complex disease. So here is a Manhattan plot produced by a consortium known as CREAM, which actually studies refractive errors. So throughout the genomes, you can have tens of genes and SNPs associated with refractive errors, right? If you zoom in here for chromosome 1, in fact, here the lead SNP is this one shown in purple, but you're talking about tens to close to hundreds of SNP that are in strong LD with this lead SNP are also possible candidates that you should follow up to study their potential functions. So to follow up the SNPs associated in GWAS, and you can use functional studies to follow up these SNPs. So we for conventional method, you have luciferase reporter assay, electrophoretic mobility shift assays, EMSA, or ChIP-seq, so on and so forth, right? But then these conventional functional studies are actually done one by one, so that's why the throughput is extremely slow. Okay, and because you have tens of hundreds or even thousands of it to follow up, in order to prioritize the work, then you have to sort of prioritize it through guesswork or through the use of limited information available in the literatures. So that's why it's actually almost impossible to follow up tens of thousands of SNP found in GWAS by conventional method. So that's why through synthetic biology, the genomic version of this method makes it possible to do pooled screening. So we have massively parallel reporter gene assay, MPRA, or single nucleotide polymorphism screening, sequencing, SNP-seq. So I'm going to spend more time on MPRA, massively parallel reporter assay. So MPRA can quantify the effects of a large number of cis regulatory elements and their variation on the expression levels of a reporter genes. So MPRA here, we're talking about you're studying a large number of SNPs or sequences that are potential cis regulatory elements, right? And in fact, MPRA was first reported in 2009 in Nature Biotechnology. So here you have to create an MPRA libraries. That libraries also contain codes, transcribed barcodes. Okay, so what are the barcodes here? In conventional reporter assay, you have a reporter gene, and in front of the reporter gene, you have the fragment of interest that may carry the cis regulatory elements. But then in MPRA, in order to study a large number of cis regulatory elements at the same time, you need to have barcodes that is linked with the particular cis regulatory elements, so that when you count the barcodes as part of MPR of the expressed mRNA, that gives you an idea of the expression level of the reporter gene under the influence of the cis regulatory elements under study. Right, so the number of barcodes expressed as RNA give you an idea of how much is the expression of the reporter gene under this effect of cis regulatory elements. And because you're going to study a large number of cis regulatory elements, that's why in the library you have many plasmids that carries different cis regulatory elements in order to distinguish different cis regulatory elements. Then you have distinct barcodes linked to each cis regulatory elements. And then the whole library of this plasmids is transfected into an appropriate cell lines and then you express the genes and sequence the mRNA. So by quantifying the barcode mRNA normalized to the input of DNA, this ratio gives you an idea how good or how much is the reporter expressed under the influence of a particular cis-regulatory element. And there are two ways to produce an MPRA library. So here you have the first approach. Actually, you order a DNA oligo pools. So in the pools, you have the fragment of interest already linked to the barcode. So the fragment of interest and the barcode, in fact, is synthesized as part of the process, right? And then the entire oligo pools is cloned into a plasmid, and then the reporter gene is cloned in a second cloning step in between the fragment of interest and the barcode. And then with the reporter gene, with this complete MPRA plasmid, then the whole library is transfected into appropriate cell lines for follow-up. And then the second approach is actually the oligo pools is produced on its own, just containing the fragment of interest without the barcode. The barcode is then linked with the fragment of interest through a separate cloning step or through a particular type of PCR procedures. And in the third step, then the reporter gene is cloned in between the fragment interest of the barcode. So here you need one more steps here. Either what we do, either you have oligo pools containing the fragment of interest and the linked barcodes, or actually you have to clone the barcodes and link to the fragment of interest in the separate steps. Okay, the same, once you have this MPRA libraries, then the libraries has to be transfected into a mammalian cell lines to study the expression. And here, because you are studying a large number of fragments of interest, so manual designing and assigning of barcodes is not actually cost effective. So that's why researchers have produced software tools for designing the fragment of interest and also the barcodes. The first software of interest is actually MPRNator. Okay, but this is actually less desirable when compared to a later online tool based on our package, which was actually published in 2018. In fact, we'll recommend this one. Although in our own group, we have our own bioinformatician writing as his own R script to do the designing. Okay, the reporter gene can be clone downstream of a medium promoter, a strong promoter. Medium promoter is actually suitable for studying enhancer regulatory elements. Strong promoter is then designed for studying repressor regulatory elements. In fact, for MPRA, there are several applications. One is to carry out differential analysis for allelic sequences, or MPRA done under different environmental contexts in different subtypes of treatments. The second application is to characterize thousands of different putative regulatory elements. The third one, if you have a few established regulatory elements and you want to study the effects of every possible mutations of every space within those few established regulatory elements, then you can use saturated mutagenesis approach and then study by MPRA. Okay. So for statistical analysis of MPRA results, that start from simple use of parametric or non-parametric tests of the expression level, t-test or Whitney U-test in the very beginning. And because of the more applications, then different research groups produce specific packages for analyzing MPRA, including this Q-ACER MPRA, MPRA score, so and so forth. In later packages, in fact, they can also include prior information using Bayesian modeling that actually helps to provide more meaningful analysis of the MPRA results. So the general observations from all these studies is actually increasing the number of replicates of the MPRA studies actually has a similar effects to increasing the number of barcodes that you include in the MPRA study. and also a small increase in the number of replicates of the MPRA studies beyond 3, in fact, give you a great improvement in the power of your study. And finally, substantial saving in cost and or experimental efforts may be achieved by appropriate choice between deeper sequencing of your RNA library versus additional replicates of your studies. MPRA is made possible because of 2 recent advancements in technology. The first one actually is the availability of DNA oligo pools, whereby you can produce a large number of unique single-stranded oligos by means of microarray technologies. In fact, you can produce more than 1 million unique single-stranded DNA in a single pool. To quantify the expression for many different barcodes at the same time, that requires the use of next-generation sequencing. Here, I'm going to say a few more words about the oligo pools. In fact, up to now, throughout the world, I think there are not more than 10 companies that are able to produce DNA oligo pools for use in MPRA. But then you have to pay attention to the following features of the oligo pools. Number one is the maximum length of oligos within the pool. In fact, this maximum length of oligos that can be produced actually varies from company to company. And the maximum length is actually up to 300 bases. I mean, in fact, the organizer is the company that can offer this maximum length of the oligos. And also the maximum of oligos in any single pool produced in a single run. My understanding, in fact, it can be more than 1 million distinct oligos per pool. And then also the yield of each oligo in the pool. And you have to pay attention to whether the amount of oligo will vary with the total number of oligos per pool. And also the error rate, okay, the lower the better. The uniformity of the oligos in the library. Of course, the more uniform, the better. Okay, and of course, because you need to use specific MPRA plasmid. Nowadays, these plasmids are easily available through AdGen. Here, our own application, in fact, we are now doing the NGS to get our result. We are waiting for the result to come back. But I'd like to sort of illustrate that, in fact, I mean, to follow up GWAS SNPs are using MPRA. 2 papers been published in 2016 back to back. One actually by Chewy, I mean, and then the other one is by Ulich. In fact, this one is actually studying the GWAS-associated NUCs that finds to be associated with red cell traits. Okay, and in fact, the culmination of MPRA is the report of studying close to 6 million SNPs in a single MPRA experiment. So this is the latest one, the most extensive one, which is published in Nature Genetics in 2019. Okay, and next, in fact, and then all these are episomal RNA because the plasmid DNA is not incorporated into the chromosome. Is based on plasmid chromosome. In fact, in this case, the chromosome context is missing. The other one is chromosomally integrated MPRA, less popular. It's actually based on lentivirus vector. Here, because of the use of lentivirus vector, then the fragment of interest is randomly integrated in the chromosomes at multiple sites, but possibly not at the native sites. The observation is MPRA in the integrated versus the episomal context are reasonably well correlated. But then the integrated MPRA results are more reproducible, more robust, and more biologically relevant. Okay, another variation of MPRA is STAT-seq, which stands for self-transcribing active regulatory region sequencing. The major difference between MPRA and STAT-seq is actually in MPRA, the fragment of interest is cloned upstream of the promoter and the reporter gene. But in STAT-seq, the fragment of interest and hence the barcode is actually cloned downstream of the reporter gene. So this is the major difference. Okay, Another genomic version of a classical functional study is the EMSA, and the genomic version is the SNP-seq, first reported in 2018. So here, protein-bound DNA oligos will be retained in a protein purification column after extensive washes, right? And then after recovery from the column, SNP-containing oligos that are bound to nuclear proteins can then be sequenced to determine the allele-dependent protein binding. So very simple. So here you have short double-stranded oligos interacting with transition factors from nucleic extract and then purified through the column, and then you harvest the protein-bound oligos and then follow up with sequencing. Okay, the sequencing which will tell you which alleles actually will bind more proteins. Okay, and that actually is the genomic version of the EMSA. Last but not least, in fact, I'd like to just mention briefly parallel screening can also be done for genome editing. So you can have multiplex genetic screening through genome editing. There are different versions here: PREDICT-seq, MOSAIC-seq, or CRISPR. These are different examples of massively parallel genetic screening based on genomic editing. So to conclude, the use of genomic tools of synthetic biology allow us to understand complex disease after the GWAS. Okay, I'll stop here and take questions from the audience. Thank you very much.